maybe even going a bit beyond and making transgenics, which all requires manipulating cells. So I hope you all have your printouts. This year is the first time that you should print this on the web page before the lecture, ideally, or prepare yourself for the lecture so that you have questions and you can ask me or the lectures to follow. Yeah, print this off the web page. It's on Canvas. Did somebody look on Canvas? Who looked on Canvas? Good, 2, there. But you actually all should look on Canvas. Have a look that you can see the handouts, have a look through, and prepare yourself for the lecture. This is how you profit most of it. Yeah? So what we're going to talk today is about cell transfections. This is just a brief overview what we're going to look into. We're going to review what are transfection techniques. This obviously deals with cells. Then we want to look into more advanced systems like microinjections or viral delivery. This is another way to bring DNA into cells or organisms. We want to review what means transient or stable transfections. We'll look a little bit into RNAi knockdown and inducible systems. And at the end, we talk a little bit about CRISPR-Cas9 genome editing and also expression tools and some new techniques that are emerging from Cas9. So transformation means that we give DNA to bacteria and they take it up. Yeah, this is use of naked DNA. Mostly when we talk of bacteria, we have a plasmid. This is what bacteria naturally have, and this is how they can exchange information. But obviously, we use it for our purposes. So we have an antibiotic resistance on them to maintain it in the bacteria. We also need to have origin of replication. And most of the plasmids we use, they are high copy number, so that we get lots of DNA. And then we have some gene of interest that we are interested in, not the bacteria. And if you want to transform bacteria, this requires competent cells. There are two types of competent cells for bacteria. One are biochemical competent cells, where we induce with heat shock, we favors the bacteria to take up the DNA. Most of these cells have been prepared with calcium chloride or rubidium chloride. There are various methods. But generally, heat shock competent cells are not really highly competent cells. We can get the plasmid in, but if you want to clone something, this might not work as well. Another method is physical transfection, and that means is we use electroporation. So we have the bacterial cells in water, and then we apply a high voltage. And because DNA is charged, it will go in one direction. And so basically, through electroporation, you shoot the DNA into the bacteria. Obviously, this is much more efficient. Now, this is how we transfect bacteria, and some of you might have already done this. But now we want to talk about transfection. What this really means is a mixture of two things. This is transformation, which you do with bacteria, and infection, which comes from viral infection, for example. It's a mixture of these two words. And it basically means that we bring DNA into cells, not bacteria, eukaryotic cells. So basically, we have nucleic acids, maybe from a virus, and this results in production of a complete virus when this virus infects a cell. So historically, it was found in 1928 by Frederick Griffiths that he could transform Streptococcus. So he had a pathogenic strain and a non-pathogenic strain, and the pathogenicity could travel from one strain to the other. So this means there was something that could be exchanged between the different strains. Then only in 1944, Avery, McLeod, and McCarthy discovered that DNA is a transforming factor. So they could isolate DNA. It was about to be characterized about that time. I mean, the concept of a gene was still far away. Or the double helical structure was not known at that time. But they found that DNA, that was known at that time, would be the transforming factor. And then in 1964, Foltz and Troutner used the term transfection for the first time. And then methods were developed to bring DNA in. And one of the first ones, described by Ryan Bagano, was the DNA dextran transfection technique, which you're going to review a bit as well. So, transfection is the same principle as transformation, but with mammalian cells. And what we want to do is to put nucleic acids into cells using either biochemical or physical processes. Biochemical methods, as I pointed out, is DAE dextran or calcium phosphate. There are liposome-mediated transfection methods. And then some more novel methods which use organic molecules with specific features, also involving magnetofection, so applying a magnetic field to bring things into cells. This is what we're going to look at in more detail. And then there are physical transfection methods, which is electroporation, I briefly touched on. We can shoot things into cells, like using little particles, like a gun, like a gene gun. And also use microinjection to basically punch a hole in cells and put things in there. These are physical methods that we all use in the lab. And then biological ones are using a virus. So for this, we need to generate a virus with our gene of interest that can affect only cells, but nothing else, and not propagate. So it's specifically designed to only carry the gene of interest, but not be pathogenic, obviously. So there are lots of different methods around for various techniques, and specifically for transfection methodologies. Have a look in this book, Current Protocols in Molecular Biology. For any types of methods, this is really a good resource as a first start for looking into a protocol. The university has access to it, so through the library you can get into, and for example they have various chapters on it, for example transfection of cultured eukaryotic cells using cationic lipid reagents. So with these methods you can make your reagents yourself, and obviously it's much cheaper than buying expensive stuff from companies. Or you have other methods, transfection UDDAX dextran, calcium phosphate transfection. So these are classic protocols that you can find that people are using for decades. And obviously, they work. And you also find a little discussion about pitfalls that might be useful. So for any type of things, this is really a good resource. So the most classic transfection methods are with calcium phosphate or DAE dextran. What they do really is to facilitate DNA binding to cell membranes. Calcium phosphate will precipitate the DNA onto the cell membrane. DAE dextran is charged, so the DNA will bind to these particles. Yeah, DNA is negatively charged, this is positively charged. They associate with DNA, the complex again binds to the cell surface. And then DNA is taken up by endocytosis to little vesicles and then are released into the cell. And the plasmid is maintained and, for example, expresses your favorite protein in cells. These methods are cheap, but for a number of cells they work quite well. But most people decide to use more expensive things, despite they work the same way. So if you do transfection, consider your method. Other transfection methods use liposomes. So these are little vesicles that are made out of lipids, pretty much like the lipid layer on a cell. So you have a lipid bilayer, which has the charged part on the outside and the lipid part on the inside. And you form this bilayer here, and in the middle you can have DNA. So they have very similar properties, chemical and physical, like biological lipids. They form spontaneous complexes with DNA. So if you mix these lipids and DNA in water, then you can form little vesicles. And they're obviously very high efficiency for transfections, because these lipids, like before, they will fuse with the cell membrane because it has the same biophysical properties, and then release the DNA into cells. There is no size limitation, like for viruses. The capsid gives a certain size that you can use. If you reach that, you won't be able to package the DNA. And obviously, they're also safer than viruses. There are lots of commercial available reagents available, like lipofectamine from Vitogen, Fugene from Roche, Effectgene from Cryogen. Every company has their own mix and claims it's better than the other company's ones. But you can also make your own mix to transfect cells. So the type of reagent depends a bit on the application and cell type. Obviously, if there are more fragile cells, you need to play around more with methods. Other cells are more easygoing, and you find high transformation rates. They're about 5 to 100-fold more efficient for transient transfection, and about 30 to 20-fold more efficient for stable transfection than the chemical methods we discussed before. But obviously the question is, do you need this high efficiency? And you might find that the effort is a bit over the top. Then the next way to put in DNA is applying the same principle as we use to transform bacteria with electroporation. But for bacteria, this is easy because they are rigid enough to put them in pure water. Because if there was salt in there and you apply a high voltage, I guess you can imagine what happens. About the same thing as if you put the hairdryer in your bathtub. You generate a shortcut. Yeah? So with bacteria, this works so well because you have them in water. But obviously cells, you can't put in water. You still need to have them in a buffer, which has salt. So this limits a bit the application of electroporation, but it does work. So the principle is that we use a high voltage to induce an electric shock. And because the DNA is charged, it will migrate. But the electric shock does something else. Because cell membranes are an insulator, current will not be able to pass. So if you apply a current, this produces holes into the cell wall. And this is the way how the DNA gets into. So if you apply a high voltage, this results in a temporary breakdown of the cell membrane and formation of pores. And this is how the DNA gets in. This is the machine that we use for this. And we have this little apparatus where we place these electrocuvettes in, and then we press the button for a short period of time, and then a high voltage is generated, and the DNA migrates. But obviously, if the voltage is too high, then we generate a shortcut, and the cuvette arcs, and the cells are dead inside. So you need to adjust the conditions for this. It needs a bit of playing around. But for some cells, this works quite well. So we have variance, which is the amplitude and the length of the electric pulse that we can adjust according to the cells. And the buffer, we have them in this. And as you can imagine, it's quite rigid, so a lot of cells will die. But the efficiency is generally good for cell culture. But this can also be applied for primary cells. So, for example, if we try to culture cells from an organism, and where the resource is not limiting or so, then this can be a good alternative. It's less affected by the DNA concentration, but there's a linear correlation between the amount of DNA present and the amount taken up. And this can also be used to make stable transfection. So obviously it's very quick. You don't need to do much. You just put the cells in the cuvette and transform them with the DNA. So another biophysical method is to use this gene gun. So literally, it's a gun. And it shoots very small particles. So to do this, we precipitate the DNA onto gold or tungsten particles. And then we can shoot the DNA into cells. So mostly this is used for cells that have thick cell walls, like plants. So plant cells is very difficult to get into because they have a thick cell wall. So with our other methods, we wouldn't be able to transfect them. And with this gene gun, we can simply shoot them in. But we can also use it for other cells, like primary neural cell cultures or so, shoot this in. It can also be used for in vivo delivery to tissue. Principally, you could hold it to your leg and shoot it in your muscle if you wanted to transfect yourself, but I don't recommend this. Then there are other methods, polyamine dendrimers and magnetofection. So these are newly developed macromolecules. They are positively charged and will bind the DNA, and obviously also have properties to facilitate entry into cells. They are highly branched molecules that will complex with negatively charged DNA. And there are various companies that produce those ones, for example, QIAGEN, and they call these superfact because apparently it's to be so good and costs lots of money as well. More recently, people have used magnetic fields and nanotechnology so that small particles are moved with a magnetic field to deliver DNA into cells. Obviously, this is very gentle and brings the DNA into cells, so you have a high survival and efficiency rate. So these are more the classic methods that we use for cells. Microinjections is more special, but is used for some cells where we really want to be sure something is in there. For example, if you want to look at cells and how they migrate or how they behave, or so, maybe then we want to be sure that they have been transfected. And maybe we don't want to wait until the construct we have transfected expresses a GFP to monitor these cells. So through microinjections, we can be sure which cells have been transfected because we include a visible dye. And the efficiency is very high. So if you're good and you inject the cell, then stuff is in there. But it's very time-consuming and requires skills. I used to do a demonstration of this, but unfortunately our equipment is broken. But for those that are still interested and want to do this, they maybe can talk to me. And we also offer projects. If you really want to try it yourself, you can talk to me about the project using this technique. Now, this is less used for cell culture cells, but is, for example, the technique for in vitro fertilization and for making transgenic animals. So if you're very skilled in this is something that is very looked for. For example, we use it in the lab to make transgenic Drosophila. That's the only way to get transgenic Drosophila. It's also popular in xenopus oocytes because these oocytes are big and you can inject them fairly easy. Either into the cytoplasm or the nucleus. This is used for studying gene expression or transport in the cell, but is also highly used for expression of ion channels and then doing patch clamp recording. So this is a way to look at proteins that are involved in regulating membrane potential. So this is a setup, is a microscope, and then you have micromanipulators on the side, and they will manipulate this needle here. Yeah? So there's a needle associated, and then you have cells in here, or Drosophila embryos, or frog oocytes. And then through this little wire, you can apply pressure, either through oil-driven or just by air. This is how you push this in, with a little pulse of pressure. So if this was a oocyte for in vitro fertilization, you can see here the needle that will inject, and then there's the second thing here, which is very big. And this is to hold the cell. This is in solution, you need to hold it. So this applies a little suction, and then holds the cell there, and then with the second needle, you can inject it. And this is actually what you see when the needle goes into the cell. For example, of a mammalian cell being injected. So it requires lots of skills. No? It's very fiddly to get these cells, but this is how in vitro fertilization is done. And also a bit how we inject Drosophila. Then there are biological methods which uses viruses. There are three main types: adenovirus, retrovirus, or lentivirus, and they have different properties. So viruses, they really develop very fancy methods, mechanisms to get into cells. And this is basically used to transfer genes for our purposes. So we use the tricks of the viruses. So we need to have a viral construct, and then also some viral genes to basically pack it into a viral particle. So the packaging of these viruses is separated from where we express the virus. So you first have to bring the DNA into cells that are specifically designed to package the virus, and then we purify it from these cells and apply it to a second type of cells, so infect them. So this way we can break the cycle of pathogenicity of viruses. So we just use the parts of a virus that it needs to package the particle from its replication. So this way we can use viruses safely. One of the popular viruses is adenovirus. This is used for transient expression mostly. So if you have just cell culture, then this is what you can use. Retrovirus will use a stable integration. So they will integrate into the genome, but targets only proliferating cells. So if you want to study neurons, for example, they won't divide anymore. And we use lentivirus. They also will result in stable integration, but only in non-proliferating cells. And some of these viruses, like adenovirus, is also used in gene therapy, for example. So, this is a viral vector. So what are key components that this vector needs? Can someone tell me?
Origin of replication.
Yeah, why do you need that?
To have a starting point.
Uh-huh.
Where you want to put the gene of interest.
Uh-huh. And where do you grow your vector? What organism do you use to grow your vector?
Our bacteria is used.
Yeah, bacteria. So every DNA that we manipulate, we grow in bacteria. This is our workhorse. Yeah, and it needs an origin of replication. What else does it need for the bacteria? Yeah, because we want to study a gene. But does this make it grow in the bacteria? What else do you need to grow it in a bacteria? So if you just have the origin of replication and your gene of interest, what will the bacteria do with it?
Just keep reporting, keep going.
Say again?
It will keep going. You need an end of replication as well, so it can be.
I'm not sure about that. What else do you need?
Some do the lack of
What do you need that the bacteria will keep your construct?
Gene of antibiotic resistance.
Yeah, and antibiotic resistance, because if you don't have that, the bacteria just eat your plasmid. Yeah? So you need a selection. So these are the two things, is origin of replication and resistance. This is what you need to grow this in a bacteria. Yeah? This is the bacterial vector. And then you have a part that makes up the virus. So this is a shuttle vector that you can use in bacteria and in mammalian cells. And it needs to have the two properties. So this is the bacterial part. It's a puck and ampicillin resistant. And then we have something that is called ITR. Any idea what this is? We have two of those. ITR. What is ITR? Inverted terminal repeats. Yeah, also transposons use something like this, and these are repetitive regions they use to integrate into the genome. For example, if you want to integrate something. And then we have a promoter in here, and a gene that we want to express, and then some information that is used by the viral proteins to package the DNA. So this makes up a viral reporter. This is the information we use. So then we put this DNA that we have generated in bacteria, we transfect into the packaging cells. These cells, they will take the DNA and put the DNA into viral particles. And these viral particles are then released from the cells. So in the supernatant of these cells, we have viral particles. These viral particles, they will not infect these cells normally. So they will just accumulate. And then we need to harvest them from these cells, determine the titer. So we need to know how many viral particles are there if we want to apply them. And then we can transfect it to the target cells. So you see, this is much more laborious than all the previous methods. But through this, we can, for example, study cells in a mouse brain. So we can inject these viruses into the mouse brain. And then we can do our studies there. So these are more complex biological systems where we use viruses as a biological delivery method. So what we mostly talked about is transient transfection. So we take a plasmid and transfect it into cells, and the cells keep it then for a few days. And we don't put the selection on in mammalian cells. Obviously, this means that not all cells are transfected, and we have a mixture. Maybe some cells have more than one plasmid DNA, others have only one, and some have none. So we get a whole range. So if you just want to express a protein, this is good. You see some cells. You can see how a protein is made. Maybe you can do co-IPs, or just check on a Western for any modifications of a protein, so something like this. But obviously, it's not quantitative in any way. So if you want to be a bit more sophisticated, you probably want to do stable transfections. This means that the DNA is incorporated in the genome. And it can still be 1, 2, 10 copies, maybe. and happens with low frequency. And in this case, normally we have also a selection on it, so that only the cells will survive that have taken up the DNA. The other part of making stably transfected cell lines is that we need to clone the cells. So we need to start from a single cell, because we want to know the copy number, so that you can compare maybe different cell lines or different constructs. So meaning, this is taking a long time to make such cells and to grow them up. There are new technologies now that we're going to touch on later on when we talk about Drosophila, that is used in Drosophila very often, but now you can also use in cells to make this process a bit faster. But it's still going to take some time. So you need to evaluate what you need for your experiment. Is this something quick where the concentration doesn't matter that much? Or if it does matter, maybe this is the way to go. So when we transform Drosophila, this means we bring a gene into the fly. And we do this by injection. And historically, we used a transposable element as the vehicle to get into the genome. So transposons, they have these inverted repeats, which we get in the virus, and the enzyme that uses these inverted repeats to integrate it is a transposase. Yeah? So everybody has transposons in their genome, and this is where it came from. So at some point, people discovered that there were strains in Drosophila that had transposons and others that didn't. And if you have transposons in the strains, normally very rapidly inhibitors evolve. And then you can't use this transposon anymore for transfections because it's deleterious. But it's part of an evolutionary process that these transposons come, amplifying the genome, and about 90% of our genome is just transposons. But obviously it's very controlled. So, when these flies that we have in the lab, they didn't have these transposons, P element, piggyback, or minus. Either they were not present in Drosophila or they come from other species. For example, from moths or other insects. And if you have the transposase, then we can use them in Drosophila. Then again, we have a bacterial vector that we can amplify the DNA in the bacteria, and we need some fly selection marker. Most times is the white gene, which makes that the flies have red eyes. So if you take a white mutant, put in a transgene with these white genes, then we can recognize the transformants by their red eyes. So very simple. Yeah? So you find your transformant, one fly in a million of white-eyed flies just pops out by these red eyes. It's a very powerful selection. Yellow gene is another one that's frequently used, or GFP more recently. So this was one of the first ways to get genes into flies or any other organisms. You can use this. But very often, depending where your gene is inserted, you have effects from the environment onto the expression of this gene. For example, if your transposon inserts in the heterochromatin, it won't express your gene. Or if it is inserted near a very strong enhancer, it will express a lot. So you can have lots of differences. So people were interested to have something more controlled. And this came from using this Phi C31 integrase, which mediates targeted insertion. And what it uses is a landing site in the genome, which is an at-B site. It's a short DNA piece. And this is placed into a transposon that was brought into the genome and put in various places. And you can choose which landing site you wanted. And then if you inject your DNA into flies with this landing site and have this integrase, then it will integrate in this predefined position. And if you have different construct, you can all insert them at the same position. Yeah?
Are you adding the landing site to the gene?
Yeah, you need to have a fly
Yeah.
that has a transposon in the genome with an @B site.
So is that already in the genome? And
That's
just
already in the
choose
genome.
one?
And then we inject in this fly.
Why can't you put the landing site on the, like, why can't you do it in one, why does it have to be 2 steps?
Because it already needs to be there
Yeah, but,
when you inject the second one.
oh, okay. So it can't just find it.
Yeah, if you just inject the DNA with an @P site, it will go nowhere. So it needs this specific sequence. So it still requires to inject DNA into Drosophila embryos. But similar systems are also now used for cell culture. So you can buy cell culture cells which have such recognition sites in the genome, and then you can integrate them, and this is very efficiently. So obviously this way you don't have to clone your cells. So it makes it much faster. And this is standardly used if we want to express gene expression levels. So this was first used mostly in Drosophila, but now it's also used in other systems. And this @P site you might have come across is what Invitrogen sells as their gateway cloning systems. So you can use the same principle to clone things and swap inserts between vectors. So I'm going to talk a little bit about RNA interference as a way to knock down genes. And obviously this will also involve transfecting reagents into cells. So RNA interference all starts with double-stranded RNA and an enzyme called dicer that chops it down to small 21 to 23 nucleotide double-stranded RNAs. Then these small pieces are taken up by the Argonaute proteins or the RISC complex. The sense strand is degraded and only the antisense is present in the effector protein, the Argonaute proteins. Then this will bind to target RNA, and if the match is complete, it will result in degradation. So this is how siRNAs work. They have a complete match. MicroRNAs work a bit different. They mostly have an incomplete match and will result not in cleavage, but interfering with translation. So translation is downregulated. So meaning that siRNAs, they can also have off-site or off-target effects through incomplete matches. So this is a bit the limitation of the technology, but it's very powerful to downregulate gene expression. So this is a little review on microRNA and siRNA biogenesis. So siRNA biogenesis, you have double-stranded RNA, and this is cut with dicer. It cuts out this piece, which has a 5' phosphate and a 3' overhang of 2 nucleotides. And this makes it distinct from RNA degradation products, because degraded RNA will never have a 5' phosphate. So just from the things you eat and the RNA is degraded from that will not make it into a microRNA, because you won't have the 5' phosphate. MicroRNAs, they come from their own genes. They have a cap and a poly-A tail, and they can be as just a microRNA or be in other genes integrated. Then what you have here is this stem loop, and this is first cut by Drosha in the nucleus. And then this part is released into the cytoplasm, and then dicer will cut off this loop here. This is how microRNAs process. And obviously, if you want to express siRNAs from vectors, they will follow the same processing procedure. So RNA interference was first described in C. elegans by Andrew Fire, and others in 1980, and only a few years later was awarded the Nobel Prize because it was so fundamental, a process that hasn't been seen before, and also the RNAs. And I'm sure you heard this week who got the Nobel Prize this year. Who did follow in this? Everybody heard about this?
Biomolecule imaging? No, that wasn't that one, was it? That was a different one.
No, but who?
Sorry.
Yeah, circadian rhythms. And the other one? It's about cryo-EM. To look with electron microscopy at proteins to get a structure. Very, very crucial technology. So this is about RNA interference. So Andrew Fire described this as being the reactive agent is double-stranded RNA. It's sequence-specific, and RNAi targets mature mRNA, so it needs to be in exons. So if you make a siRNA against an intron, it will not work, because that's in the nucleus. And RNAi is in the cytoplasm, so it's post-transcriptional. The target mRNA is degraded because it's cleaved. Then a few molecules are sufficient, so this means there is some amplification mechanism that leads to spreading, meaning that RNA is systemic, so that it can spread between tissues and progeny. And some of you might already recognize this probably is very specific to C. elegans and doesn't apply, for example, to mammals. So there are a few things that we need to consider when we look at high eukaryotes. So double-stranded RNA is the active molecule. It's true in Drosophila and C. elegans and plants, but not in mammals. If you take long double-stranded RNA, this will trigger an interferon response. So this is an antiviral response that basically leads to cells to die as a defense against viruses. So also long double-stranded RNA is a good tool in invertebrates and plants. It can't be used in mammalian cells. So there, only 21 to 23 nucleotide small RNAs can be used to avoid this interferon response. Then a few molecules are sufficient. So the amplification of double-stranded RNA, this is only present in plants and C. elegans and requires an RNA-dependent RNA polymerase. Similar enzymes are present in Drosophila and humans, but this amplification mechanism is not. So it won't become systemic. This is also present only in plants and C. elegans to be efficient. So if you want to use RNAi in eukaryotes, we need to use 21 nucleotide small RNAs. So how to make long double-stranded RNA? This is very simple. We design PCR primers with T7 promoters in it, and then we amplify a piece of DNA. A T7 promoter is a short 20 nucleotide sequence that is recognized by a bacterial polymerase that you can buy. And it's a polymerase, it will recognize the sequence and then transcribe. So if you give a T7 promoter, T7 polymerase, nucleotides, it will make you RNA very efficiently. And it does this from PCR products as a template. So this is very efficient. And then we can transfect, for example, Drosophila S2 cells, or feed it to C. elegans, and we have RNAi. So this is such a T7 promoter. This is the sequence that needs to be here, and the polymerase will recognize this. The important part is that you need to have 3 Gs here. It won't initiate without 3 Gs, or very inefficiently. This is the limitation, but normally such sequences can be found in the genome, or be just extra sequence. We can also chemically synthesize RNA. There are companies that do this, like IDT or Dharmacon. And then we can get short RNAs. They become anneal, and you can transfect them with things you buy. Synthesis of RNA is expensive. So one nucleotide, I think, is about £5 currently, while for DNA we pay 5p. So, it's much more expensive, 100 times more expensive. So this will give expensive experiments, so we need to design them very well. And obviously, we use for other methods to do this. This is the protocol, how to design siRNAs that you can look up. So another method to make siRNAs is to clone them in front of a U6 promoter, which is a very strong promoter. And then we can put them on a plasmid and transfect them into cells. It's much cheaper. So how we do this is we have this U6 promoter. We still need to start with a G, and then we have the siRNA. We use a little spacer that makes this loop here. And then we have a U-rich sequence, which is the termination signal to make just a short RNA. And so what we end up from this is we have this in the genome. This is what was cloned. No? You can make this as 2 oligonucleotides and clone this in your vector. Then this is transcribed to make this RNA in the nucleus because of the sequence. So there's no Drosha cleavage needed. It's just how it is designed. Then dicer will come and cleave up this little loop. And here we have a perfect double-stranded RNA from a plasmid. And you can see when you apply this, in cell B, you get good knockdown, 80% or so. This is what you can achieve. Between 50 to 90% knockdown. So it means you're not removing the gene completely, you're just reducing its function, which obviously has implications for your experimental design. Then, if we do RNAi experiments, we need to consider a few controls. Scrambled siRNA can be one, or better is the mutated siRNA, so that you put in mutations. However, this could act as a microRNA because it has an incomplete match and still impact on translation. So obviously this is what needs to be checked. A better way is to use additional siRNAs that target the same gene but different sequence. So then you should get the same result to claim that this is gene-specific. The empty vector obviously is not a control because it doesn't express an siRNA. And there is a reference dealing with off-target effects. So obviously this is something that has to very critically be taken into account, that siRNAs with an incomplete match might act as microRNAs and have impact on gene expression. Obviously, we want to claim that when we knock down specifically a gene, that the phenotype we see is because we knock down this gene. For Drosophila, there is a transgenic library available. Here lists 20,000 genes, 20,000 inserts, but meanwhile probably there are around 40 to 60,000 lines available with siRNAs, which you can order from stock centers. So a lot of tools are available for this organism. And what they use is these siRNAs are hooked up to a US promoter from yeast. And this a promoter that is activated by Gal4. So we have one fly line that has the siRNA, and then we cross them to another line which has a tissue-specific promoter. For example, that expresses only in the muscle or only in the brain. And when we cross these flies together, we can study the knockdown in a specific tissue and see what the effect of this is. So this is a very powerful tool to look at gene knockdown in specific tissues and to circumvent viability problems. So if your gene is, for example, required for viability, then you wouldn't be able to study this because embryo might be embryonic lethal, and you would never get to the adult to study its effect on neurodegeneration, for example. But these are ways to deal with this complexity. So it's a very good tool. Another system that we often use in cell culture systems, also in transgenic systems, is that they need to be inducible. So mention, for example, if you want to do an RNAi knockdown, and this gene has a very fundamental role, it will kill the fly or the organism. So we need to be able to induce this and study the effects more controlled. This is what we use inducible systems for. The classic system is called TET on-off. This works fine for some acceptation, but it has problem with leakiness. So if this is an issue, then this might not be the right system. Leakiness means that it'll express a little bit, not much, but a little bit. But if, for example, the protein you try to express is toxic, it'll just kill the cells and you never get something. So you need a tighter control. There are some novel systems that use steroid hormones. For example, this one uses 20-hydroxyecdysone, which is only present in invertebrates, but not in vertebrates. So this is a naturally evolved system that has very tight controls. And because it is not used in vertebrates, it is of benefit. And the same is progesterone can be used in invertebrates because they use ecdysone. Ecdysone is an insect molting hormone. There is no toxicity in vertebrates for this. The same is true for this progesterone analog, RU486, which happens to be used as abortion pill as well in humans, but is used in insects to induce gene expression. So how the system works is the classic one is the TET off system. So we have TET transactivator, and normally this one binds to the promoter, the TET-O promoter, and will induce gene expression. Now, if you add doxycycline, this will bind to the DNA binding protein, the TET-R, and will make that it falls off the DNA and will not be transcribed. So this is the classic situation. Then this was turned around so that normally it wouldn't bind the TET-O promoter, and only if you add doxycycline, we will get transcription. So this was a mutated TET-A activator that binds the promoter. So on paper, this all looks good, but practically it has some limitations because the control is not as tight as it looks on paper. But for many applications, this works quite well and can be used. If a really tight control is used, maybe this 20-hydroxyecdysone system is of use. So here we need vectors that contain the receptor dimer, which is the ecdysyl receptor and another protein called RXR. And they bind together to this response element. Now, if there is no ecdysone, they will not recruit the transcription machinery. If you provide ecdysone, then this will bind to the receptor, and this will recruit the transcription machinery. And then we get transcription. You can see here in cells that without the transactivator, there is no expression, while with the transactivator, you can see GFP. So this way, we can decide when we want to do the gene expression. This is a way to timely control this, and also a little bit to control the amount that is made. Not a lot, but there is a small range to determine the amount that is expressed. This is a reference to look up this protocol. Another method that is prominently used, particularly in Drosophila and also mice, is using a clonal analysis. There are various ways to use this, but in whatever flavor it comes, it uses small sequences like these FRT sites. They come from yeast. The vertebrates mostly use LOX-A sites, but there are many others that can be used. These are short recognition sequences for a recombination event. So recombinase will use these sequences. So what we use this here is to make a rescue construct. So we have a promoter that expresses in neurons where this gene normally is expressed. And so we made transgenes in a mutant background. And then it will express in all neurons, for example, because it's neuronal. Now, we can then make clones by inducing this recombination enzyme, FLIPase, with a heat shock. So there's a heat shock promoter. When we briefly increase the temperature, we'll express these genes. But it will express this very little, and only a few cells will express them, not all. And then we can get these clones. No? The protein EWG, where we have an antibody against, is absent. Then we use the trick to visualize this clone. So we clone the GAL4 yeast transcription factor. And so if this cDNA is removed, then the promoter will drive GAL4. Generally, eukaryotic genes are monocystronic, so a promoter drives what is downstream, but will not express this because there's a poly-A site. Huh? Only if this recombination happens, then the GAL4 will express. And then this will activate UAS gene, which drives GFP. So we can visualize the clone. So this comes in many different flavors, but it's a prominent way to express or manipulate genes in a specific area. We can also have these FLIPases with specific promoters and direct this to specific tissues. This is often used in mice to, for example, knock out a gene in specific cells. So this is a really prominent technology that is used. So the last 15 minutes, we're going to talk about CRISPR RNAs. This is something that has come up in the last few years and is really a technology that has revolutionized many applications because it uses nucleic acid sequence to target a DNA scission enzyme to make a specific cut. This can be used for downstream manipulation of DNA. So it's a technology to edit DNA. CRISPR stands for clustered regularly interspaced short palindromic repeats. And what this all is an RNA-based bacterial antiviral immune defense system. So bacteria have evolved this system of incorporating pieces of DNA from phages into their genome, and then they express them and can recognize the phages. A bit similar like the siRNA system, which is also an antivirus defense system. So the viral DNA has been incorporated into the bacterial genome and then becomes expressed as short RNAs. These are the CRISPR RNAs. And they direct cleavage of viral DNA by Cas enzymes or enzyme complexes. So this is the principle of it. And bacteria use this as an immune defense. So, for example, if this virus comes here, then if this bacteria had exposure to virus, then maybe this sequence had been integrated in these repeats. And these repeats are expressed into small RNAs and processed to have a constant part, which is the orange part, and a variable part, which is this, the variable part is the orange one, and the constant part is the loop. So this is the guide RNA. This is taken up by the Cas complex, which can be one or several proteins. And because this is a DNA sequence or RNA sequence that will recognize another sequence, it will have a complete match and can distinguish the DNA from the bacteria from any invader. Yeah, and if it has a complete match, then it will cleave the DNA of the phage. And that's it for the phage. So it will not propagate. So this is the defense mechanism. That the DNA on the first exposure has been incorporated into the genome, and then it can be transcribed to make this sgRNA, or this short RNA, and this can be used for the defense. There are different Cas enzymes, and there are more and more of those being discovered, but generally they form into three groups. One is type 2 enzymes. This is the one that's most commonly used. This is just a single protein that has all the features in it. Type 1 have 2 proteins, and type 3 have 3 or more that form the active complex. Obviously, for manipulating DNA in vitro, or how we want to use this, type 2 is the most prominent one. And then they also have some different requirements for the RNA that is used. So if you talk about Cas9, then this is a type 2 enzyme. One single enzyme that will incorporate a single-stranded RNA and then make a double-strand cleavage. So historically, Cas9 required 2 RNAs for activity, the CRISPR RNA and the tracer RNA. So you can see here, this is the CRISPR RNA that hybridizes to the DNA, and then the tracer RNA would hybridize to the CRISPR RNA to this constant part. And the tracer RNA is required for Cas9 activity. So it's a way to tightly control this powerful enzyme, which is obviously relevant for bacteria, but maybe not for us when we want to manipulate DNA. So what we use is what's called sgRNA, which is a short guide RNA, which is basically what is shown here in green. And then this is fused to the CRISPR RNA. And in practical, this end is also fused to this 5' end of this RNA, so there's a little loop in here, so that we can express all as one RNA that will form this structure and be functional. A name that you come across frequently is called protospacer, is this part where the RNA hybridizes. So this is the target region. Then for the specificity, Cas9 needs a minimal sequence requirement, and this is 3 Gs, 1 nucleotide after the end of the hybridization part. So this NGG is the protospacer adjacent motif. So there need to be 2 G nucleotides for Cas9 to be active. And then this complex will cleave the double-stranded RNA to produce blunt double-strand breaks. So this machinery is a way to induce double-strand breaks into DNA. And it does this on both strands. You know, so the RNA is only hybridizing to one strand, but the cut will be a double-strand break. So this is the fundamental tool to produce double-strand breaks. Specifically use double-strand breaks, and nearly every position we can use this to edit our genome, or any genome. And this is using double-strand breaks and homologous recombination. So if you have a double-strand break in our genome, either this is fixed in a so-called SOS repair, which is put together to make sure the chromosomes don't fall apart. But it's not so good, but the cell has this ability. And so if a double-strand break happens, you can find mutations. Or, most of the time, this is fixed through homologous recombination, where there's a template. So normally, we have 2 chromosomes. No? And if a double-strand break happens in one chromosome, the second chromosome is used to repair the other one. This is why we have 2 chromosomes in the cells, to repair it. So if a double-strand break happens, we use this template, which is the second chromosome, or a template that we provide. And then, for example, we can put in a mutation in this template, or even a piece of another gene, GFP, for example. And then it can be incorporated and result in an edited genome. So if you can produce a double-strand break, then we can manipulate the genome. So before, this was not really possible. Yeah?
Beforehand, you said that there were blunt ends. How do they stick? Because I know with sticky ends, they just bind on, but you said that there was blunt
Yeah, if there were sticky ends, this would aid to fix them, because there's a little overlap. But they just produce blunt ends, because the normal way to repair this is via homologous recombination.
So how do you get your stuff in if they're not sticky?
Oh, you know, one strand will invade the template and be synthesized on the next strand. So classic
when it
homologous
repairs, it just
recombination.
incorporates your foreign DNA.
So if you, let's go through this a bit more detail. So if you have a double-strand break, no, then you have these 2 DNAs, and then you have a nuclease that will recess one end and make this single-stranded. And so if you get this single-stranded end, it will then intervene with your template and be synthesized on this template to make it longer.
Okay.
And this way, you will synthesize the gap that is present here. So if you're not entirely sure, you might want to take a book
I think I might look.
and look into this to really understand what is happening here. Basically, use a template to synthesize the next strand, and then this one will fall back onto this template to synthesize the rest. And this way, you fill this gap. So this is the principle of homologous recombination. Cas9 can also be used, for example, to cut plasmids. And we developed a method in the laboratory to manipulate large plasmids for cloning using a similar method, using homologous recombination in bacteria. It's called recombinering. If plasmids get big, they're very difficult to manipulate. But by using these homology arms on the side and producing a gap in a plasmid, we can fix this with homologous recombination in bacteria. Obviously, this requires that we find restriction enzymes that can make such a gap, which you still find in 20 kb. You can find enzymes that cut once, but obviously there is some sequence limitation. But now we can use Cas9 to cut a plasmid, as shown here. Yeah, this is with restriction enzymes, we'll cut out a piece, and this is with sgRNAs, and Cas9 is in vitro cleaved plasmids. And we've used this to clone a mutation into this plasmid. And with this method, yeah, you can see the green ones are the recombined ones, 100% efficiency. All clones have this. So it's a very powerful method. and much superior than classic cloning methods. We have this in revision. Hopefully it's going to be published soon, then you can all read it up and use it yourselves. There are some theme variations to sgRNA Cas9s, and that is there were Cas9 enzymes engineered and that are cleavage dead. And then this can be used, for example, to control transcription. So we can have a transcriptional activator and bring it into a promoter to act on pol 2. So simply by this sgRNA that hybridizes, sequence specifically, we can bring in new regulators. So here, Cas9 will not cleave, it will just sit there. And the purpose is exactly to just sit there and bring in an activator, for example, to activate transcription of a specific gene. Or it can be used to label DNA in vivo. If you fuse it to a GFP, we can bring GFP to a locus in the DNA. So these are just two of the emerging new applications for Cas9 together with sgRNA, but there is more. So it's good to watch out for new developments in this field. And the latest was this one, which was last year. It's a very popular research field, so there's lots of development. So watch out in journals like Cell, which is the most prestigious journal, to look out for new developments in the fields. So just want to summarize this part. So sgRNA and Cas9 is a sequence-independent DNA editing tool. So we can determine the specificity by designing where the sgRNA is going to hybridize. Gap repair recombination is highly efficient and robust with highly copy number plasmids. So we can use this homologous recombination also in bacteria to manipulate plasmids. And this is very efficient. sgRNA Cas9 system can efficiently cut plasmids for gap repair recombination, but the efficiency depends on GC content and sgRNA structure. So a downside of sgRNAs is that is a bit temperamental. Not all sgRNAs work, and there aren't any clear rules, but they can be used for in vivo genome editing, regulating gene expression or labeled DNA. But the targeting rules so far are a bit diffuse, so this requires more research to also make this technology safe for genome editing. So there's ongoing research in this field. Another application that can come out of this is gene drive systems. There's basically a self-perpetuating knockout construct. So you can use this to eliminate a species. And obviously, making such things is strictly regulated. So don't attempt. Okay, that's from my side. Any questions?
Please summarize inducible systems.
The principle is that you have a way to control the expression. So mostly this is a small molecule, like a hormone. And for this you need a transcriptional activator that will only be activated in the presence of this hormone. So it's a classic hormone response. So you have a transcription factor that does nothing. And only when the hormone is there will induce it. Heat shock is another inducible system. So there are promoters that will only be activated if the temperature goes up. This is the principle of it. So that we can control when we want to express a gene.
Excuse me, can you suggest some good books, if you don't come from this kind of background?
Well, this lecture is assembled from many different areas. I think if you're specifically interested in one or the other topic, I'm sure there's more literature to it. Just go on PubMed or Encyclopedia of Life Science to find more literature on it. Methods paper is something to look for if you're specifically interested in methods. The current protocols, if you're looking for protocols, this is a good resource to get more about a specific method. But also reading primary research paper, and particularly the methods part, helps to understand this. And lastly, try it in the lab. That's mostly how it works out. At some point we have to try it and do it ourselves, and then we figure out how it works. Anything else? Good? Then we'll stop here.